16.3 Cell Transport. Learning objectives: explain how passive transport works, explain how active transport works. Every living cell exists in a liquid environment. One of the most important processes carried out by the cell membrane is to keep a cell in homeostasis. Homeostasis is a state of relatively constant internal physical and chemical conditions. It does this by regulating the movement of molecules and other substances from one side of the cell membrane to the other side. The cytoplasm consists of many different substances dissolved in water. The process by which particles move from an area of higher concentration to an area of lower concentration is known as diffusion. Diffusion is the driving force behind the movement of many substances across the cell membrane. In this instance, the area of higher concentration is outside the cell membrane. The area of lower concentration is inside the cell membrane. Diffusion depends on random molecule movements. Therefore, substances diffuse across the cell membrane without requiring the cell to use additional energy. The movement of molecules across the cell membrane without using cellular energy is called passive transport. Since cell membranes are built around lipid bilayers, the molecules that pass through them most easily are small and unchanged. These properties allow them to dissolve into the membrane's lipid environment. Proteins in the cell membrane act as carriers, or channels, making it easy for certain molecules to cross. Red blood cells, for example, have protein carriers that allow sugar glucose to pass through them in either direction. These channels facilitate or help the diffusion of large glucose molecules across the membrane. In facilitated diffusion, molecules that cannot directly diffuse across the membrane pass through special protein channels. Hundreds of different proteins allow substances to cross cell membranes. Although facilitated diffusion is fast and specific to certain molecules, it is still diffusion, so it does not require any use of cell energy. Water molecules cannot easily diffuse through the cell membrane. However, many cells contain water channel protein known as aquaporins that allow water to pass right through them. The movement of water through cell membranes by facilitated diffusion plays a role in an extremely important biological process known as osmosis. Osmosis is the diffusion of water through a selectively permeable membrane. In osmosis, as in other forms of diffusion, molecules move from an area of higher concentration to an area of lower concentration. The only difference is that molecules moving in the case of osmosis are water molecules, not solute molecules. So osmosis is the diffusion of water through a selectively permeable membrane. Water will tend to move across the membrane until equilibrium is reached. At that point, the concentration of water and sugar will be the same on both sides of the membrane. When this happens, the two solutions will be isotonic, which means same strength. Note that strength refers to the amount of solute, not water. When the experiment began, the more concentration sugar solution on the right side of the tube was hypertonic, or above strength, compared to the left side, so the diluted sugar solution was hypotonic, or below strength. Driven by the differences in solute concentration, the net movement of water out or into a cell produces a force known as osmotic pressure. Osmotic pressure can cause an animal cell in a hypertonic solution to shrink and one in a hypotonic solution to swell. Because cells contain salts, sugars, proteins, and other dissolved molecules, they are almost always hypertonic to fresh water. As a result, water tends to move quickly into a cell surrounded by fresh water, causing it to swell. Eventually, the cell may burst like an overinflated balloon. In plant cells, osmotic pressure can cause changes in the size of the central vacuole, which shrinks or swells as water moves in and out of the cell. Fortunately, cells in large organisms are not in danger of bursting, because most of them do not come in contact with fresh water. Instead, the cells are bathed in blood or other isotonic fluids. The concentrations of dissolved materials in these isotonic fluids are roughly equal to those of the cells themselves. Isotonic, the concentration of solutes in the same inside and out the cell. Water molecules move equally in both directions. Hypertonic, the solution has a higher solute concentration than the cell. The net movement of water molecules out of the cell causes it to shrink. Hypotonic, the solution has a lower solute concentration than the cell. The net movement of water molecules into the cell causes it to swell. Although diffusion is useful, cells sometimes must transport materials against the constant difference. The movement of materials against the concentration difference is known as active transport and requires energy. Think of a child running up a slide. It takes tremendous energy to run up a slide. The active transport of small molecules or ions across a cell membrane is generally carried out by transport proteins, or protein pumps. They are found in the membrane. Larger molecules and clumps of material can also be actively transported across the cell membrane by processes known as endocytosis and exocytosis. The transport of these larger materials sometimes involves changes in the shape of the cell membrane. Energy from ATP is used to pump small molecules and ions across the cell membrane. Active transport proteins change shape during the process, binding substances on one side of the membrane and release them onto the other. In endocytosis, the membrane forms a pocket around the particle. The pocket then breaks loose from the cell membrane and forms a vesicle within the cytoplasm. In exocytosis, the membrane from the vesicle surrounds the material, then fuses with the cell membrane. The contents are forced out of the cell. Endocytosis is the process of making material into the cell or taking material into the cell by the means of infolding or pockets of the cell membrane. Phagocytosis is a type of endocytosis in which extensions of the cytoplasm surrounding a particle and package and then enfolds it within a food vacuole. The cell then engulfs it. White blood cells use phagocytosis to remove damaged or foreign cells and destroy them. Amoebas use this method for taking in food. Many cells take up liquid from the surrounding environment in a process similar to phagocytosis. During a type of endocytosis called pinocytosis, tiny pockets form along the cell membrane, fill with liquid, and pinch off and form vacuoles. Endocytosis is when many cells release large amounts of material. During exocytosis, the membrane of a vesicule or vacuole surrounding the material fuses with the cell membrane, forcing the contents out of the cell. The removal of water by means of contractile vacuole is one example of a kind of active transport.